Hello everyone, my name is Omar Tamim. Inshallah, in this video I'll explain the second part of the immune chapter, which mainly includes the adaptive defense and the pathologies related to our immune system. So starting off with the adaptive system, which we know is the body's built-in specific defense system, and it mainly has like three features: that it is specific and systemic, and at the same time it has a memory. So what we mean by being specific, that it has specific lymphocytes or specific immune cells that react against specific foreign substance or particular pathogens. And being systemic, that the immune response that is provided is not restricted to just one side, it is mainly systemic. And for memory, that our immune system will recognize this pathogen or foreign substance later when we are re-exposed to this same foreign substance again, and it will mount then a stronger immune response against this pathogen. So we have mainly two types of adaptive immune responses. First one, which is the cellular immunity, also known as cell-mediated immunity, and we have the humoral immunity, which is also known as antibody-mediated immunity. And from its name, we can say that the cellular immunity or cell-mediated immunity, this immune response mainly provided by immune cells. And at the same time, this type of immunity mainly targets infected cells such as virus infected cells, parasite infected cells, cancer cells, and also cells of foreign graft. So many of these cells are infected, and they act by mainly two ways, which they either kill these infected cells or they release some chemicals that either enhance the immune response or activate some immune cells. While for the humoral immunity, or what's known as antibody-mediated immunity, the immunity is mainly provided by antibodies, and humoral mainly means the body's fluid, which are the blood and the lymph, and these antibodies will circulate in this body's fluid, which are the blood and lymph, and they target extracellular the, the antigens or molecules, which might be bacteria, bacterial toxins, or free viruses. So they act against extracellular foreign antigens that are circulating in the blood, not like the cellular immunity, which are infected cells. And they act by targeting these foreign antigens. They either inactivate them or mark them for destruction by either phagocytes or by enhancing complement activity. So let's make ourselves familiar with some terms, which are, first of all, antigens. Antigens, they are mainly proteins that are recognized by our immune system as foreign. They are recognized mainly as non-selves. So we have some antigens that are presented on our body cells that are self-antigens. These are not recognized by our immune system, so our immune system won't react against these. And also we have two types of antigen. We have the complete antigen and incomplete antigen. So the antigen should have two important functional properties, which are immunogenicity and reactivity. Immunogenicity, which is that this antigen has the ability to cause a lymphocyte to proliferate or increase in number or multiply, and the activity, that this antigen should be able to react with these lymphocytes that proliferated. So complete antigen has these two functional properties, while the incomplete antigen only has immunogenicity. It only has activity without immunogenicity. Unless it binds to a carrier protein, then it becomes a complete antigen. Also, the incomplete antigens are known as haptens. We also have other terms, which is antigenic determinant. Antigenic determinant are part of our antigen that are recognized by our immune system. So immunogenicity mainly depend on this antigen term and depends on the size and the complexity of this antigen. So the larger the size and the more complex these antigenic determinants are, the more the more this antigen is immunogenic. And we have also another term which is self-antigen, which are proteins or antigens that are presented on the cell membrane of our cells that are not recognized by our immune system, or they are recognized as self and not foreign. We also have a term which is MHC proteins. These are mainly proteins on the cell surface that has a groove where antigen is presented on it to our immune cells. And as we can see here, these are the MHC proteins, and the dark brown ones are the antigens that are present on the MHC protein. So the three main cells that are involved in the adaptive immune system are they have a T cells, the APCs, and the B cells. So now for the lymphocyte development, maturation, and activation. Lymphocytes are white blood cells, and white blood cells at their early stages are hematopoietic stem cells that originate from the bone marrow. So lymphocytes, they originate from the bone marrow. After they originate from the bone marrow, the B lymphocytes, it stays in the bone marrow for maturation, while the T lymphocyte, it goes to the thymus to be mature. So the aim of maturation is to develop immunocompetence and self-tolerance. What does each one of them mean? Immunocompetence, that these lymphocytes will react against a specific antigen, so it will be for only a specific antigen. So here it gains its specificity. And self-tolerance, that these lymphocytes won't recognize our self-antigens, so they won't react against them. So it mainly, the maturation it acts like a protective mechanism against any autoimmune disease. Following the maturation, seeding these lymphocytes, they go into the secondary lymphoid organs. What are the secondary lymphoid organs? These are the organs in where these lymphocytes gets activated, they get activated, proliferated, and they act against the foreign antigen or pathogens. So we mainly have the primary lymphoid organs where lymphocytes are produced and they become mature, which are the bone marrow and thymus, and secondary lymphoid organs such as the spleen and the lymph nodes. Following, so when they enter into the lymph nodes and the spleen, they circulate for any antigens. Following that, a clonal selection occurs where these antigen binds to the receptors of these lymphocytes. So the antigens are the ones that select lymphocytes. Why? Because as we know, these antigens are proteins, and on the lymphocyte there are receptors, and mainly receptors are the ones that binds to these proteins. The proteins are the one that binds to these receptors. So this antigen selects these lymphocytes. When it's selected, it binds to it. Following this binding, proliferation and differentiation of the lymphocyte occur. So the lymphocyte that the antigen binds to it's mainly called the ancestor cell or the clone. This cell proliferate and differentiate into two main types of cells, either the effector cells or the memory cells. So as I said earlier, that the immune cells of the adaptive defense, they only recognize a specific antigen or a pathogen. And our immune system mainly recognize hundreds and thousands of pathogens. So how does this happen? It's mainly due to the antigen receptor diversity. And antigen receptor diversity is mainly achieved by a process known as somatic recombination. So as we know, the receptors are proteins, and how proteins are produced mainly by transcription translation. So, so in our bodies we have mainly more than 20,000 genes. So these segments of these genes are shuffled and they recombine again to form these receptors. So that's how our body produce diverse antigen receptors. So this slide is mainly excluded, but I only included just to deliver the main concept of maturation, which happens in two steps. First of all, positive selection, then negative selection. So in positive selection, they test these T cells. A type of cell in the primary lymphoid organs are known as thymic cells. They mainly test these T cells. First of all, they check if they are able to recognize our self-MHC. If they were not able, they will go under apoptosis, which is also known as cell death. And in the negative selection, these thymic cells check if these T cells are able to recognize self-antigens. If they were able to recognize them, they will also be killed because they must produce a T cells that won't be able to recognize self-antigen. Because if they were produced, they might act against our self-antigens, leading to autoimmune diseases. So we said that one of the cells that are involved in the adaptive defense are the APCs, or which are known as antigen-presenting cells. So these cells, what they mainly do, they engulf the antigen and they present these antigen on the cell surface to our immune cells for it to be recognized. We have three main APCs, which are the dendritic cells, we have the macrophages, and we have the B lymphocytes. First of all, the dendritic cells, what do? They engulf this antigen and they migrate to the lymph node to present these antigens to the T cells. So T cells will be activated. And B lymphocytes, they mainly present these antigens to helper T cells. So these helper T cells will activate the B lymphocytes, and the same goes to macrophages. These macrophages will present these antigens to naive T cells, which are helper T cells. So these T cells will activate these macrophages. These macrophages are already functional, but they activate more. It's more like an upgrade for these macrophages. So in humoral immunity, or which is known as antibody-mediated immunity, the activation and differentiation of B cells occur by two steps, which are the clonal selection and then the proliferation and differentiation of these B lymphocytes into effector cells and memory cells. So in clonal selection, these antigen binds to the receptors on the B lymphocytes. Then following that, these B lymphocytes proliferate and differentiate into two cells, which are effector cells, and also they differentiate into memory cells. They differentiate into effector cells, that is the plasma cells, which is the antibody secreting cell, and they secrete antibodies or produce antibodies to target the foreign antigens. And for the memory cells, these memory cells are produced in case we got re-exposed to the same pathogen again. Our immune system will recognize this pathogen faster and it will elicit a stronger immune response against it. So we know a thing that's called an immunological memory, which is occur after the first or the primary immune response. So we have two main types of responses. We have the primary immune response and secondary immune response. Primary immune response, it is the immune response that occurs at the first exposure, while the secondary immune response, it occurs after the first exposure. It can be the second, it can be the third, it can be the fourth. In the primary immune response, the antibody titer peak may reach after 7 to 10 days, while in secondary immune response, it will reach after 2 to 3 days. And in the primary immune response, the memory cells are produced in this response, while in secondary immune response, it depends on the memory cells produced by the primary immune response. And secondary immune response are faster, it lasts longer, and they are more effective and last longer than the primary immune response. And in humoral, in the humoral immunity, in primary immune response, the IgM antibody is mainly produced, and at the end IgG, while in secondary immune response, only IgG antibodies are produced. Yeah, mainly it's IgG. So as we can see here, a on the first exposure, the antibody titer peak reaches after 10 days. What we mean by the antibody titer is the antibody concentration, while in the second immune response, it reached after maybe 2 to 3 days, and it produced more antibodies. As we can see, the concentration of antibodies were higher. So we have two types of humoral immunity. We have the active one and the passive one. In the active one, the B lymphocytes they get activated and they proliferate and differentiate. When the passive, the B lymphocytes of our immune system, they are not activated, and ready-made antibody are introduced in our body. So each of them, they either can be natural or artificial. For the active, natural can be by getting infected with a virus, a bacteria, or any type of pathogen, while the artificial can be by vaccination, where where the immune system will be activated and there will be only milder symptoms, or sometimes it can be asymptomatic. And vaccines are mainly like a weakened virus or a weakened pathogen or a dead pathogen that is immunogenic and reactive. While by getting natural active humoral immunity, we might get some symptoms. And for the passive, they also can be natural and artificial. Natural, these antibodies they are delivered from the mother either to the fetus or to the infant by either transfer placenta, or for the fetus or for the infant by the mother's milk. And for the artificial, we mainly antibodies are injected into our bodies, such as in cases of like when you are bitten by a snake, when you got rabies, because these uh, pathologies are mainly deadly, or these infections are mainly deadly, and it causes death very fast before the immune system gets activated. And because in active humoral immunity, our immune response gets activated, memory B cells are produced, so there will be immunological memory, while in the passive there won't be immunological memory because our immune system is not activated. For antibodies, first of all, antibodies are made up of heavy chains and light chains. These are the heavy chains. The ones are in orange or black, and the ones in blue are the light chains. And we have two regions: the variable region, which is different for which differs between one B cell and the other, and we have the constant region, which is the same for the same class of antibodies, and they are connected together by a disulfide bond, and these antibodies they are proteins. So we have five classes of antibodies. We have the IgM, IgD, IgE, IgA, and IgG. Starting with the IgM, which is a pentamer. It has 10 binding sites, 10 binding sites, as you can see here. They are produced in the primary immune response. They activate complement, and at the same time they can be a B cell antigen receptor. We have also here the IgA. IgA, it's a dimer. It has four binding sites, as we can see. They are found in the body's secretions such as sweat, intestinal juice, and milk, and also saliva. They they act against any pathogens that attach to the epithelial surface, so they protect the mucous membrane and the epidermis. And also we have the IgD, which is they're not merely a true antibodies because they do not bind to extracellular molecule or pathogen and they inactivate them. They just act as receptors on the B cells. So their main function, they are B cell antigen receptors. And we have the IgG. The IgG is the most abundant one. They are produced in the secondary immune response and the late primary response. And they also activate complement. And the last one we have is the IgE. The IgE, they binds to mast cells and basophils. So whenever an antigen binds to these antibodies, they cause the release of histamine and some chemical mediators also that might lead sometimes to allergic reaction. So how our antibodies mainly work? A antigen-antibody interaction occur. They either inactivate or they trigger our immune response by either neutralization, agglutination, precipitation, or complement activation. So neutralization occurs when all the bindings, all the antigenic terminals on a single antigen, they are blocked by antibodies, and this bacterial toxin or the bacteria or the virus becomes inactivated and they can't perform their main function. Or agglutination occur where mainly IgM is the one responsible for agglutination. Agglutination because they are a pentamer, it has 10 binding sites. So agglutination occur when a single antibody binds to multiple antigens. They agglutinate, and this will enhance phagocytosis. And precipitation is similar to agglutination, but this happens for soluble antigens. And the last one is complement activation. The binding of these antibodies causes the activation of the complement, and what the complement do, they cause cell lysis, inflammation, or phagocytosis, and the neutralization, agglutination, and precipitation, they also cause phagocytosis. They target these antibodies so the phagocytes or the macrophages can target them and destroy them. So in this homeostatic imbalance, so as I explained how does antibody works, a sometimes against some parasitic worms that are very large, this antibody interaction doesn't work on these. So what happens that these parasitic worms will be caused by IgE antibodies, and whenever we are affected with and with these pathogens, eosinophils will bind to the stem of the IgE, which might lead to the release of histamine, and it triggers and it causes inflammation. So cellular immunity or cell-mediated immunity is not included, it's excluded, but I'll talk about it briefly. So we have three types of cells that are involved in cellular immunity. We have the helper T cells, cytotoxic T cells, and regulatory T cells. The helper T cells, they are effective CD4 T cells. They either activate immune cells. These immune cells can be B lymphocytes, cytotoxic T cell, and other immune cells. They activate them by either binding to them or by releasing some cytokines. We have the cytotoxic T cells. These T cells mainly cause destruction of virus-infected cells, cancer cells, grafts. We have the regulatory T cells. These regulatory T cells, they protect or prevent any autoimmune disease, so they inhibit self-reactive T cells. And the activation and differentiation of the T cells occurs by occurs in two steps, occurs by the presence of two signals. The first signal is the binding of a TCR to the MHC that's holding the that's holding the antigen, and the second signal by the binding of what's called the CD20 to the B7. And the absence of the second signal might cause the cell to go into energy, where the cell will be won't be reactive anymore. So it, the second signal sometimes it's not present the when these dendritic cells or the APC binds to a self-reactive T cell. So it mainly protects against any self-reactive T cell or autoimmune disease. So yeah, as I said, the absence of the co-stimulatory signal, which is a secondary signal, might lead to energy. So we have two types of MHC proteins. We have the class 1 and class 2. Class 1 is mainly present in all nucleated cells. So it's present in all cells except the RBCs. And the class 2 MHC protein, they are present only on the APCs, which are the dendritic cells, macrophages, and B cells. And the antigens that are presented on the class 1 MHC proteins, they are recognized by only cytotoxic T cells, and these antigens are mainly intracellular antigens or endogenous. And for the class 2, they are recognized by helper T cells, and these antigens are mainly are antigens of extracellular pathogens. So sometimes in cases of heart failure or kidney failure, an organ transplant is needed. But when this organ is introduced in our body, this organ might be recognized as a foreign by our immune system. So our immune cells might start to react against these tissues or organs and start to destroy them. So in cases of allograft, when a tissue or an organ is transplanted from so allograft mainly means that this graft is transplanted from different individual but from the same species. So when this allograft is done, we mainly give immunosuppressive therapy in order to uh, suppress our immune systems so they won't react against these graft cells and destroy them. And before any transplant is done, a blood typing is done, ABO blood grouping is done, and other tests. So to ensure that an immune response won't occur or only a mild immune response might occur. And immunosuppressive therapy will be mainly given after the surgery. So I just wanted to shed some light on some clinical terms that and as I remember one of these terms came in our TBL in our immune system TBL. So there are mainly only three clinical terms, which is autograft, isograft, and xenograft. So sometimes a tissue graft is transplanted from a single tissue in our body to the other. So the tissue graft is transplanted from a certain location in our body to the other location, but they are only from the same person. So this is known as autograft. We have the isograft, where this tissue is transplanted between two identical individuals, or what you call as twins. And we have also the xenograft, where tissue is taken from an animal and it's introduced in our body. For example, in cases of a when our heart valves become insufficient, a heart valve will be transplanted from the pig's heart into the human heart sometimes. So our immune system is associated with some pathologies, and one of these pathologies is immunodeficiency. So immunodeficiency is a condition in which our immune system is either impaired or it's defective, and this occurs due to failure in the production or the function of these immune cells or complements or antibodies. And we have two types of immunodeficiencies. They either can be congenital or acquired. Starting with SCID, which is also known as severe combined immunodeficiency. It is a congenital immunodeficiency. It's caused by a genetic defect that leads to the production of defective B and T cells. So there will be mainly a defect in the B and T cell responses. And if we do not treat this condition, it might lead to death, Elshaeir, and the hematopoietic stem cell transplantation has showed to improve the survival rate of children born with SCID. We have the Hodgkin's lymphoma, which is acquired. It's also known as the cancer of B cells. So B, non-functional B cells will be produced and they will occupy the lymph nodes, leading to a swelling of the lymph node, and they depress the lymph nodes. And as we know, the lymph nodes are the secondary lymphoid organ, which is site of is the B and T lymphocytes activation and differentiation. And this condition can be treated by immunosuppressive drugs and chemotherapy because it's caused by cancer. And we have the AIDS, which is acquired immunodeficiency syndrome. It's mainly caused by a virus called HIV virus. So what's special in this virus? It's the only virus that has an enzyme called a reverse transcriptase, and this enzyme has the ability to convert RNA into DNA. So it turns into a provirus. And these cells target mainly the helper T cells. So when they target the helper T cells, these helper T cells gets infected, and the helper T cells has a specific important significant role in our immune system. They are the cells that activate these cytotoxic T cells, they activate the B cells, they activate macrophages also, yeah. So, and this condition, it can't be cured, but we can manage it in order to increase the lifespan or the quality of life of anyone gets infected with this virus. We also have autoimmune diseases. Autoimmune disease occurs when our body fails to recognize uh, our self-antigens as self-antigen, and they think that these antigens are foreign. They start to mount an immune response against them, and antibodies and cytotoxic T cells will start to destroy our own body's tissue. So we have seven autoimmune diseases. We have the rheumatoid arthritis, in which a, the joints will be destroyed by our immune system. We have the myasthenia gravis, in which antibody will be produced against the acetylcholine receptor in the neuromuscular junction, so they mainly interfere with the muscle contraction. And we have the multiple sclerosis, in which the myelin sheath that surrounds that traps the axons gets destroyed. And we have the type 1 diabetes, in which it's caused by the destruction of pancreatic beta cells, which are the cells that's mainly responsible for the production of insulin. We have also glomerulonephritis, in which it results in renal impairment and the kidney's filtration membrane will be damaged. We have the systematic lupus erythematosus, in which this disease might affect mainly the kidney, heart, lungs, and skin. And last, we have the Graves' disease, in which antibodies will be produced, and these antibodies affect the thyroid gland to produce excessive amount of thyroxine. So these autoimmune diseases, they are treated by different ways. First of all, an autoimmune disease, there will be an inflammation. To suppress this inflammation, we give anti-inflammatory drugs, for example, corticosteroids. We also have some therapeutic approaches and some other therapeutic approach, but they are still under development. But but the ones that are used, we use antibodies to block the action of cytokines or the receptors of cytokines, because cytokines enhance inflammation. So we block the action of these cytokines. We also block the second signal that we discussed that activates the T cells. So if you block this signal, cell will go into energy and it won't be responsive. We have also some therapeutic approaches, but they are still under development. We either activate regulatory T cells, because regulatory T cells will inhibit self-reactive T cells. We also induce self-tolerance using a vaccine, and we destroy the self-reactive cells by using antibodies against them to target them and destroy them. So the failure of self-tolerance occurs mainly due to a defect in the action of Tregs or the co-stimulatory signal. And also there are some other mechanisms that lead to the loss of tolerance. For example, when a foreign antigen resembles a self-antigen. For example, in rheumatic fever, an antibody will be produced against the bacterial infection that caused this rheumatic fever, but this antibody will also bind to some of our self-antigens and cause destruction, because it somehow has similar structure, so these antibodies can bind to these antigens. And also by the appearance of new self-antigens that our immune system is not aware of. For example, due to a gene mutation that leads to the production of new proteins. Or, for example, when a hapten or incomplete antigen binds to our antigen and the structure of this antigen changes, to our immune system will recognize it as a foreign. Or sometimes there are some self-antigens that our immune system is not aware of. They are hidden behind barriers, for example, behind the blood-brain barrier. And in case of trauma, these antigens will be exposed, so our immune system will react against them and destroy. So the last disease or pathology or condition associated with our immune system is the hypersensitivity. So hypersensitivity is when our immune system gets more sensitive and gets somehow out of control and starts to destroy ourselves due to a perceived threat that is not harmful. So we have four types of hypersensitivity. We have the type 1, type 2, type 3, and type 4. These types of hypersensitivity, they differ in their time course, and the first three types, they involve antibody, while the type 4 hypersensitivity only includes T cells. And we have the type 1 hypersensitivity, which we know as an allergy. It's also called the immediate hypersensitivity or the acute hypersensitivity. So this type of hypersensitivity causes an allergic reaction. How does this allergic reaction? So on the first exposure to this antigen, there won't be any signs of allergic reaction, because at the first exposure a sensitization occurs where the basophils and the mast cells will be caught by IgE. So on the second exposure or subsequent exposure, the antigens or these allergens will bind to these receptors and they will cause the release of histamine. So this type of hypersensitivity can be localized or it can be systemic. The localized one, which we know as the allergy that causes vasodilation, runny nose, watery eyes, and itching. It also sometimes, if it enters through the respiratory tract, it might cause what is known as asthma, where the bronchioles gets constricted and the airflow gets restricted. And if it enters through the GI, it might lead to GI discomfort. While in systemic, these allergens gets into our blood and they circulate our whole body, and they might lead to what is known as anaphylactic shock. And this anaphylactic shock might lead to death because it causes bronchoconstriction, and at the same time they cause vasodilation that leads to circulatory collapse. So this anaphylactic shock leads to restriction of airflow and difficulty in breathing. At the same time, it leads to circulatory collapse, and it can be managed and treated by epinephrine. We have the subacute hypersensitivity, which is the type 2 and type 3, and type 2 is also known as cytotoxic hypersensitivity, and type 3 also known as immune complex hypersensitivity. It's supposed to be 3, not 4. So in type 2, what happens is these antibodies binds to an antigen on cell surfaces, and they lead to stimulation of phagocytes, and they cause cell-mediated lysis. It mainly occurs after mismatched blood transfusion. When blood transfusion occur, antibody will be produced against these RBCs, and they will bind and cause cell lysis. So antibodies binds to an antigen on cell surface. While in the type 3, antigen antibody complex will form, and these antigen antibody complex are insoluble, so they will deposit in the cell membrane, and this will be followed by complement mediated lysis and cell destruction by neutrophils to get rid of these antigen antibody complex. And these reactions or responses, they also lead to the destruction of the surrounding tissues and cells. An example of type 3 hypersensitivity is glomerulonephritis, or what's of the SLE. And the last type of hypersensitivity, which is the type 4 one. The type 4 one is mainly caused by T cells, unlike the type 1, and type 2 and type 3, which are caused by antibodies. In the delayed hypersensitivity, inflammation and tissue damage are caused by the cytokines produced by helper T cells that activates macrophages, or by cytotoxic T cells that directly cause the destruction of these T cells. An example of delayed hypersensitivity, we have the allergic contact dermatitis, which is caused by direct contact of chemicals. These chemicals will diffuse through our skin, binds to self-proteins, and they will be recognized as foreign, and our immune system will mount an immune response against it, and they will destroy it and destroy its surrounding tissues. And so that's it, and thank you.